Okay. Up next, Rachel Gross from Plant Science.
So close your eyes for a moment with me and imagine you're walking through a meadow and you see hundreds of flowers in bloom. This beautiful scene begs the question, how does a plant know when to flower? As scientists, if we can understand how plants flower, we can use this knowledge to breed new cultivars of plants that flower earlier, resulting in a quicker product to market. Along with understanding flowering time, for other crops such as tomatoes and peppers, there's another important factor in cultivar development, and that's fruit characteristics. The fruit of a tomato can be highly variable, and different characteristics lead to different markets. This is evident just by going to your local grocery store and seeing the tomatoes that are available. Some are very large, like the beefsteak, and other are very small, like cherry tomatoes. Both fruiting and flowering characteristics are controlled by a combination of environmental cues and genetics. Now, every cell in a plant contains the same genetic data. However, only some of that genetic information is actually helpful for fruiting and flowering. Typically, these genes involved are also tightly regulated so that they're only turned on during the correct developmental stage of a plant's life cycle. Unfortunately, there's no single thing that controls gene expression, but rather a combination of different tools. First, we must understand how genetic data is packaged into a cell to begin to see the complexity of gene expression. DNA is tightly wound around protein bundles, and these are called nucleosomes. The, the nucleosome arrangement and modifications are one aspect that can affect gene expression. There are some special tools that eukaryotes use to help manipulate these nucleosomes to modify gene expression. One of these tools are called chromatin remodeling complexes. Chromatin remodeling complexes can shift the placement of nucleosomes and modify the structure of the proteins inside them, and by doing so, it can have a direct impact on gene expression by either increasing or decreasing the accessibility to a gene. We have found a chromatin remodeling complex called SWR1 in tomatoes that directly affects flowering time and proper fruit development. When we remove an essential component of this chromatin remodeling complex, the tomato plants have delayed flowering time, and the fruits produced are very small. They have low levels of sugars and fewer seeds. These results suggest that SWR1 is responsible for gene expression of key flowering genes and fruit development genes. Our future work is to investigate precisely which genes SWR1 is targeting to alter their expression. By understanding this complex process, we can make better crops to help feed the world more efficiently. Thank you.
Thank you. Fascinating. I love tomatoes. So
Good, so do I.
this is interesting. So by working in this area, do you hope to then develop some tomatoes that then could go to different parts of the country based on different latitudes, things like that, that would have different flowering times?
Preferably, it's actually more desirable to eliminate any sort of like light requirements for flowering, just because then your product can be grown in any region and isn't just like restricted to one area over another.
Excellent. And how long do you think it'll be before you actually see this commercialized?
That is a good question. I don't know. We need to first figure out which genes are being controlled by this complex, and then we'll kind of go from there.
Okay, fascinating. Thank you.
Yeah, thank you.